Hi students, good morning. And today I'm going to show you how to search, filter, and make a library of lead molecules against a selected drug target using freely available databases such as PubChem, Kimball, Drug Bank, and Mining databases. There are so many databases are available for you, but many of them are paid. Some of them are partially accessible by the academicians and researchers by using their which means official mail ID. And here, these I have given these databases are freely, easily accessible and freely available for you at any time. So you can use this for your project, right? And your aim is to identify and retrieve the potential lead compounds because you are going to report us with the list of compounds which are capable to work against or work on the selected target proteins, right? So lead molecules means nothing but a drug-like substances are similar to the drug or a drug already used or are in the market, and then maybe a new chemical moiety which can be used or which can be capable of acting as a drug towards the selected target protein, right? So you are going to choose that list of compounds and submit whatever you listed, filtered, right? Introduction part and materials required you can read whenever you have time because we are running short of time. I will go directly to the protocol. In the protocol, we have 5 different steps. In the first step, we need a biological target, right? So you have to select a precisely well-defined target. If it is has a prot ID and 3D structure in the PDB means that is well and good. Easily our job will done. Then search of after the target determination, you can go for uh, searching of known ligand standard inhibitors of the particular target. What is available drugs are available inhibitors are available markers which are have the proven activity. Okay, those are called as known ligands. And bioactive compounds means here so many labs, research labs around the world have the research, right? And they have deposited the results against the target. So we can get the bioactive compounds also from the databases. For that, we are going to search the chemical structure, right? So you can use PubChem and Chembl. One more is there. I forgot the name. Chem Spider. Okay. So that also one of the good database you can access Chem Spider also. For the practical purpose, you can use these to do very fast. It means we are going to complete, right? So PubChem databases, you can just go there and enter the protein name, target name, or gene name, whatever you have as a target in which area you are going to work or you already selected, right? In your case, you already have the protein for the entire practical, so you can, if you want, you can check that. Otherwise, you can choose any pathological pathogen protein target proteins. Okay. So in that PubChem, you can just directly go to the bioassay and select the compound against IC50 and EC50 values. So I will show you how to do that because in this protocol, uh, small things will be missed because how much value is best in IC50. A lower value is best. So for example, less than or equal to 10 micromolar of drug substances are best. And here also, this is inhibitory concentration, and this is effective concentration, right? So inhibitory concentration, both are uh, useful for us. Suppose if you want to inhibit a protein or inhibit a microorganisms through the protein means you can use IC value. Suppose some substances may have effective concentrations on that, so you can get it. Okay. After that, when we are searching, we can have list of documents list of uh, chemicals, right? So you can download as a Excel, and here that is called as CSV file, comma separated value. So you can use this Microsoft Excel to open this file. You can download it. And the next one is Campbell, mostly used database. And here also you can go and select by using UniProt ID of the protein, right? Uh, don't give protein name. If you don't know the UniProt ID means you can go for protein name. Otherwise, it is best to give the protein ID, the UniProt ID. Then select the associated compounds. With that you can here also you can sort the compound by using IC50 and Ka50. K is a constant, I say inhibitor also inhibitor constant. That also that should be very less, right? So if the drug is inhibited in a very small amount, which means in the respective to concentration means good drug. For example, if you have two different substances, one is inhibiting that particular microorganism with 10 micromolar, and another one has the 10 mg, means which one is best? 10 micromolar concentration is best, right? Because we need only minimal amount of drug to inhibit the pathogen. So don't forget this if you are selecting by using the IC50 or EC50 or Ka values. Okay, here also you can download the CSV file by using that contains the Smiles, Kimball ID, and bioactive values. Bioactive values are nothing but these IC50, EC50, and Ka values, sometimes pH also, with the values, cell line values. Okay, in the third step, uh, perform similarity search. In the first steps, we have target, precise target, and second step, we have the biomolecules which are all acting against the target. Now we are also selecting, we are going to select any similar molecules are there in this database, right? So like drug, like the inhibitor, like whatever the compound we selected, chemical compounds, right? So xenobiotics, and use drug bank to find the approved drugs if you have. If any approved inhibitors or drugs against our selected target proteins means we can get it through the drug bank. And PubChem similarity search is useful, and Zinc database, another important database you can use for mostly in real world docking. We can get the we can filter by using the Zinc database, right? Then very easiest one is Swiss similarity. So we are going to check any, we are going to give any molecule, chemical molecule, and we can check that how many similar molecules are here, right? So we can get and we can download everything from these databases. So collecting of molecule is over. After that, so in your now in your hand you have target and list of collection of the chemical molecules. Then we are going to filter the chemical molecules, which means by using the Lipinski fire rule, by using Swiss ADM. Swiss ADM has everything to filter the drug components. Only thing is you have to format the smile format of the drug. Okay, you will see that. And I hope you already knew that what is meant by Lipinski fire rule. Apart from this, you can use solvent access area. Okay, pan interference are there. I will show you when we are in the practical session. Okay, molecular weight should be less than 500 kD, less than or equal to 500 Dalton, and log P value should be less than or equal to 5. 3 is optimum. If you have more number of uh, chemical moieties and you want to filter further, means you can use 3. And hydrogen bond should be less than or equal to 5, and hydrogen bond acceptors are will be more, should be more for a drug moiety. It is possibly uh, act as an drug, effective drug, right? So you can use this. Apart from this, I have told you solvent access area and interference and so many docking also is there. At final, you have to do perform a docking, right? And here, document of the results, you should create the spreadsheet, which means Excel sheet to filter the compound, and you can discuss with that.